All right, guys, I'm going to try to explain DNA replication in under 10 minutes. Here we go. So you should be able to explain this using all the vocabulary that's here, including this list of enzymes and proteins, as well as Okazaki fragments and deoxynucleoside triphosphates. I really do encourage you to basically do what I'm doing and try to explain the process in words out loud. Okay, so you should draw it out in your notes, label your notes, but then also try to explain it in words. It's really helpful. And so these are the main enzymes, or some of the enzymes and proteins involved. You need to know DNA gyrase, which relieves tension in the DNA molecule as it's being unzipped and unwound by the helicase. So the helicase is doing what I just said. It's unzipping by breaking hydrogen bonds, and it's also unwinding the double helix. That creates strain in the molecule, and that strain is relieved with the DNA gyrase. The topoisomerase there is another word for DNA gyrase. You don't need to know that. DNA primase adds RNA to the DNA strand. It adds an RNA primer, which is needed before DNA polymerase can add DNA to the growing strand. Single-strand binding proteins help to keep the two strands apart after they're separated by helicase. Okay, so how are DNA nucleotides added? When I say adding nucleotides, that's really a short form of saying adding nucleoside triphosphates, because free-floating nucleotides in the cell are in a slightly different form than the nucleotides that are in the DNA molecule. The free-floating nucleotides have 3 phosphate groups attached to the deoxyribose sugar instead of just 1 when they're part of the DNA molecule. And so what happens when the nucleoside triphosphates come in, they find their complementary nitrogenous base. So in this case, you can see that the T, the nucleoside triphosphate with thymine as the nitrogenous base, is coming in to its complementary base, adenine. And that happens automatically without an enzyme. But then to create the covalent bond between the sugar of the strand, the nucleotide that's already attached to the strand, and this phosphate of the nucleoside triphosphate that came in, an enzyme is needed, and that's DNA polymerase, specifically DNA polymerase 3. And so the DNA polymerase 3 catalyzes this formation of a bond between the nucleotides on the growing strand, and when it does that, 2 phosphates are removed. Notice that nucleoside triphosphates are always added to the 3' end of the growing strand. Study this image and make sure you can see that. Let's go through step by step how DNA replication happens. We'll start with the leading strand. So the leading strand is created continuously, and nucleotides are added to the growing strand in the direction of the helicase. And so the leading strand is formed in the same direction as the helicase is going. Okay, so the helicase is unzipping and unwinding, and then DNA polymerase 3 is adding nucleotides in that same direction. And that's always in the 5' to 3' direction on that growing strand. Okay, so that's always a bit confusing. The first step, though, is to add an RNA primer. DNA polymerase 3 doesn't know where to start without that primer. And so DNA primase comes in and adds an RNA primer, which is a short section of RNA that's complementary to the DNA on the old strand. And then DNA polymerase 3 comes in, and it starts at the RNA primer, and it adds the free-floating nucleotides, which remember means free-floating nucleoside triphosphates, to the 3' end of the RNA primer, and it just follows helicase, and it keeps adding new nucleotides on that growing strand. That's the leading strand. It's as simple, not that simple, I know, but it's as simple as that. Okay, on to the lagging strand. So if we go to the other side, okay, since the DNA molecule, the 2 strands of the DNA molecule are anti-parallel, they're going in opposite directions. On the other side, the growing strand has to be formed in the opposite direction that helicase is going. And so that sort of creates an issue, in a sense, because it can't be created continuously. You can't just start somewhere and follow the helicase. You have to start somewhere, move away from the helicase, and then sort of jump back, make another little piece, and then jump back and make another little piece again. Those little pieces that are formed are called Okazaki fragments. And each little piece is made in the same way as the leading strand. And so if we go through this step by step, you can see first that DNA primase adds an RNA primer, and it's forming it in the 5' to 3' direction. But this time on the lagging strand, that direction happens to be going away from the helicase. Then DNA polymerase III adds the free-floating deoxynucleoside triphosphates to the 3' end of the RNA primer, and then it just keeps adding those in that direction. When it reaches the primer that was added before, then it will stop. Okay, the DNA polymerase III will stop, and so you'll have a little fragment of DNA between two RNA primers. That's called an Okazaki fragment. And that just keeps happening. Okay, so the helicase is unzipping and unwinding, and DNA primase is adding RNA primers, and then DNA polymerase III is adding Okazaki fragments between those primers. Now the primers need to be removed. The one primer on the leading strand needs to be removed, as do all the primers on the lagging strand. And that's done with an enzyme called DNA polymerase I. Okay, so what DNA polymerase I does is it removes each RNA nucleotide and replaces it with DNA nucleotides. After that's done, there's still one last covalent bond that has to form between the two Okazaki fragments. That's a sugar phosphate bond, and that bond is formed with the help of the enzyme DNA ligase. And so that's the last enzyme that's involved in this process. All right, we did it. Under 10 minutes. There's a little summary showing the leading and lagging strands at the same time. So it's your turn. You should now try to just do what I did. See if you can explain this process. See where you get stuck. Okay, if you're getting stuck somewhere, that means you don't quite understand it, and maybe you need to study it a little bit more or ask some questions to me in class or tutorial. Okay guys, good luck as you're studying this. Thanks for your patience in waiting for this video, and we'll see you back in class. Bye-bye.